the challenges and opportunities are, regardless of the disease. So that sort of, what I do is ALS, so that gives it sort of a grounding. But I'm going to talk in the beginning about some theoretical ideas about biomarkers as well. I've been a paid consultant on advisory panels to Clean Nanomedicine and Denali Therapeutics, and I have research funding from numerous organizations as well as a few industry partners for ongoing trials that we're involved in. So I'll start with kind of a good definition for biomarkers. I think most people have heard this, but in 1998, the NIH Biomarker Definition Working Group sort of came up with, I think, a good definition. The other thing that's important to understand about this is that there was already a working group on this 20 years ago, and that was sort of the culmination of their work. This has been a big focus of what NIH and FDA have been working on because of the importance of biomarkers in drug development and in understanding diseases. So I think this definition really still strikes a very important note, which is a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. And it's a broad definition, but it does sort of frame what a biomarker is. It also underscores why we need to be more specific about this as well. And so from that definition, we can sort of begin to put together a conceptual framework for biomarkers. And what I'm going to talk through now really comes from, you can find it in a number of different documents, the FDA NIH Working Group BEST document, Biomarkers, Endpoints, and Other Tools. It's meant to be a resource. And it's also outlined in the Framework for Defining Evidentiary Criteria for Biomarker Qualification. I'm not going to talk about qualification really in this talk because it's a very complex process, but I will allude to it in at least one slide. To say biomarker is just a gross oversimplification of sort of the richness of biomarkers that are needed for drug development. So in light of that, these groups have broken down into sort of further categories. Susceptibility biomarkers indicate a risk of developing a disease. Diagnostic establish the diagnosis. The definitions get a little more complicated here. Prognostic biomarkers actually predict how the disease will progress, so they're making a prognosis for the disease. Predictive biomarkers talk about the likelihood to respond to a given therapy, and I'll give some examples of why that would be important or how we might use a predictive biomarker. Pharmacodynamic biomarkers demonstrate target engagement for a therapy and may or may not be related to a disease itself. Disease monitoring biomarkers demonstrate progression of a disease over time. And then safety measure the extent of toxicity during a clinical trial. So some of these are related to disease. Some of them are not at all related to disease. All of that is covered in the definition from the beginning, but it's important to understand that, it may be really valuable to have a biomarker that really has nothing to do with the disease that you're studying when you're putting together a clinical development program for a novel therapy. I think the first question that you want, if you're thinking about developing a biomarker or employing a biomarker, I think the first question to ask is, why do we need a biomarker? And here I'll sort of walk through some of these different kinds of biomarkers and why we might need them in ALS. And I'll say at the outset, ALS has very few, if any, biomarkers, and we need biomarkers in almost all of these cases. There are some diseases where these biomarkers exist, and it's actually harder to make a case for developing new biomarkers unless they really have great characteristics compared to what exists already. So in susceptibility, we now have in ALS gene and gene-directed therapies that are in development. They're in trials now. We're treating people with AAV-delivered gene therapy with antisense oligonucleotides. And if we want to know who will soon develop a disease, we need a biomarker for that. But that would allow us to treat people pre-symptomatically and potentially head off a disease, prevent it from starting. So we might be able to identify people who have a known gene. We can do that already. The problem right now is that we might identify a population of people who have a gene that causes ALS, perhaps SOD1, so a mutation in SOD1 causes ALS, but it doesn't cause ALS to begin at any particular age. So in order to run a trial, we would have to follow people for presumably 30 or 40 years to know whether we had prevented them from starting from beginning the disease. Instead, we need a biomarker that changes before the onset of symptoms, so that when we see the change in that biomarker, we can then initiate a program of treating with these gene-directed therapies or gene therapies, and potentially see what's happening to that biomarker, and also try to head off the disease from starting. Diagnostic biomarkers seem very straightforward. In ALS, it's really still a clinical diagnosis and one of exclusion at that, so the diagnostic delay is about 12 months. If we could diagnose earlier, we could then potentially enroll in trials earlier when therapies are more likely to show benefit. In other diseases like Alzheimer's disease, actually diagnostic biomarkers are important because a substantial portion of people enrolled into trials clinically don't have the relevant pathology. It's not Alzheimer's disease, it's another dementia. So there are lots of different reasons that a diagnostic biomarker might be important, and they'll vary, but I think asking the question, why do we need this, is a really important starting point. Prognostic biomarkers, again, these are biomarkers that tell you about how the disease will progress over time. ALS is highly heterogeneous, like other neurodegenerative disorders. If we could predict the rate of disease progression, the pathway of progression throughout the body, so will it start in the bulbar region and then go to the arms and then the legs, or vice versa, we could begin to stratify trial participants, and because we're taking out noise, sort of differences between patients, or at least accounting for that in our statistics, we could reduce trial size and duration and save a lot of time and a lot of money on these trials. Predictive biomarkers are also, I think, critically important in ALS. So ALS is likely heterogeneous in cause. There are 30 different gene mutations that can cause the small fraction of ALS that's familial. They're involved in different pathways. That's clear evidence that there are sort of different ways to get to ALS. Our putative therapies that we're testing work on different pathways. So, some are anti-excitotoxicity, some are anti-inflammatory. If we could select patients that had more pathology in a given pathway, we might be able to match them to drugs that are more likely to meet with benefit in that subpopulation. And that's true of many, many disorders in neurology. And I think this kind of biomarker really could get us a long way towards matching patients to a potential therapy and improve our trials. Pharmacokinetic biomarkers are not one, there isn't sort of an NIH carve-out for this, because a lot of times pharmacokinetics is really just measuring the drug level, but I wanted to put it in here as we talk about why do we need a biomarker. Drug levels to show absorption, peak concentration, and elimination are important, and the in various compartments is a really important point. In ALS, we've done a lot of trials where it turns out afterwards that the drug never really made it in any notable concentration into the central nervous system. So we haven't even really tested whether the drug works or not. So I think PK is really can't be underestimated as at least a starting point in understanding, are you getting to where you need to go. It also helps determine for future trials dosage, dosing schedule, route of administration, etc. And works really hand in hand, and maybe as a kind of pharmacodynamic biomarker. So pharmacodynamic biomarkers is a big category for disease. So, for biomarkers. They really help determine if your therapy is reaching its intended target, how long that effect lasts, what's the dosing necessary to affect the target, and is there an effect on disease biology. Pharmacodynamic biomarkers need not have anything to do with the disease that you're studying necessarily. They might have to do with one of the biological pathways. They might be really separate and more about drug effect. And I'll talk in a second about target engagement, which is one kind of pharmacodynamic biomarker. It works together with the PK biomarker to help you determine things like dosing schedule and what's the ideal dose to take forward. And I think perhaps most importantly, it helps de-risk the next step forward. So if you can work out a pharmacodynamic biomarker, show that your drug reaches pharmacokinetically relevant levels in the central nervous system, and then show that there is a biological effect of your drug, then it really has gone a long way to making a later stage trial make a lot of sense. And companies, granting organizations are much more likely to get on board. In fact, I would say that at this point in most neurodegenerative diseases, it's getting very difficult to carry drugs forward without some kind of a pharmacodynamic biomarker early in development. Target engagement is the one that we talk about the most, I think. This is really a particular kind of pharmacodynamic biomarker. It's a marker that's very proximate to the therapy's target. So This is the receptor that you're engaging. This is the molecule that you're binding. This is, the actual target that your drug is hitting. And that's really important to answer the question, are you acting on your intended target? There are other kinds of pharmacodynamic biomarkers that are further downstream and may actually be related to disease biology that would tell you not only are you hitting your intended target, but is that target having an effect on the biological pathway you mean to affect. So if you have a drug that's supposed to interfere with CD40 ligand, which we have in ALS, then you can look at receptor occupancy of CD40 ligand, but you can also look at activation state of regulatory T cells down the line, and you might even look at something like neurofilament, which is a marker of the biology of ALS, and those would all be pharmacodynamic biomarkers at different times. So really a kind of a broad stage. I think, just as we're getting past the point of being able to say, oh, I'm going to study biomarkers, that sort of isn't enough specificity. Probably to say we have a pharmacodynamic biomarker isn't enough either. You really need to kind of explain more about what kind of pharmacodynamic biomarker that is. Another big category of biomarker is a disease monitoring biomarker, and that is really a biomarker that tells you about progression of the disease. So in ALS, phase 3 trials use clinical outcomes that can be subjective and noisy, and they are themselves disease monitoring biomarkers. These are things like vital capacity and ALS-FRSR. If I have time, I'll talk about them more. The ALS-FRSR is a functional rating scale. It's a 12-item questionnaire, 48 points, and subjective, but still better than other biomarkers or endpoints that we've been able to sort of identify in ALS. With more quantitative biomarkers, though, of disease progression, we might get an earlier indication of biological effect of therapy on disease, so really acting as kind of a downstream pharmacodynamic biomarker. A marker of disease progression that's more reliable or just less noisy. It may not be more responsive, but maybe there's less noise than our current clinical measures. And as I said, this category of biomarker really spans the gamut of kind of biomarker. So it might be a biofluid biomarker, it might be electrophysiology, imaging, improved clinical outcome measures like quantitative strength measuring, digital phenotyping or quantitative medicine, or even patient-reported outcomes. All of those would really fit into the category, but we think about them quite differently. So this is a sort of a very big category. I think it's fairly clear what this could do for trials in all neurologic diseases. And safety, I think, really, the one thing to say about safety biomarkers is that I think previous therapies that were tested in ALS, and really, frankly, in a lot of neurology, were quite safe, as opposed to, say, oncology, where, they have very toxic therapies that they're testing. But now, in neurology and in ALS, we're moving towards therapies that are more risky. So cell therapies, intraparenchymal and intrathecal therapies that are changing the immune system in really profound ways, antibody therapies, gene therapies. I think these are all introducing more risk, and we really need to be able to track safety well. Always was a good idea, but more important now. I said I wasn't going to talk about qualification of biomarkers, but this is the one piece of the sort of framework for biomarker qualification, and that's a process for saying this biomarker is good as an endpoint, and we should be able to approve drugs based on it. I think this is a framework that is really important to think about, even as you begin to think about how to apply biomarkers or biomarker discovery. And as I said before, you really need to start with the need statement. Why do I need this biomarker? What drug development program will it go into? What part of the disease am I trying to understand? And if you have a good, clear understanding of that, you'll be able to write a very clear message about why this biomarker is important. And then there's something called context of use. And that's really where you begin to think about, is this a biofluid biomarker? Is it an electrophysiology biomarker? What question are you addressing? How does the type of biomarker that you're choosing help you address that question so that you sort of get the answer that you need? And that leads then into sort of what's the benefit to the patient? Can you diagnose more sensitively? Can you be more selective for a clinical trial? Can you make that trial shorter? And also, what's the risk to the patient? Do you need to get spinal fluid? Is there a biopsy involved? And some will contain more risk than others. There is also the theoretical risk of, what if it's a false positive or negative, and does that open or close doors to patients? So I think putting that together gives you a very solid case for why this is the right biomarker to try to develop further or to put into your trial. And then you come to kind of the last questions that you ask, which are statistical, and how does it perform, and what's the assay like, and is it repeatable, and what's the coefficient of variability, et cetera, et cetera. Those are kind of the details that you work out. I think for some time we were working out those details without thinking much about how we would use biomarkers. And it became clear that we were sort of developing biomarkers that weren't necessarily going to move the ball very far down the road for ALS clinical trials or drug development, or even understanding the disease. And that ended the era of just saying we're going to explore biomarkers, and really got us into a place where you have to be much more specific about it. So I would recommend this as a great document to sort of, not so much because you'll be working on biomarker qualification, but rather because it helps you understand biomarker applications. As I said, biofluids, electrophysiology, imaging, quantitative digital medicine, clinical outcome measures, and patient-reported outcome measures are all categories that we think about for biomarkers across the board. In ALS and many other neurodegenerative diseases and some neuroinflammatory diseases, we're thinking about neurofilaments. These are the scaffolding upon which the neurons are built, and they're spilled into the CSF and into the plasma when there's neuronal injury. So they're not specific to any disease, but they do go up in a number of diseases. Urinary P75 extracellular domain is a biomarker I'll talk about a little bit later. There are numerous spinal fluid markers of neurotrophic factors, inflammatory factors that we're beginning to incorporate into trial designs. And that brings up, how do we deal with getting spinal fluid in trials repeatedly? And I'll talk about that for a moment as well. Electrical impedance myography is an example of an electrophysiology marker. This is a imperceptible current that runs through the muscle and tells about the health of the muscle and therefore the health of motor neurons. It's being used in ALS as well as muscular dystrophies. It's really being developed to the point that it's basically ready to go into clinical trials, though there are still a few questions about it, and I think it would remain a secondary outcome in those trials. For the first time ever in ALS, we can use PET scans to visualize the disease. We can see astrogliosis and microgliosis in the motor cortex. I'll show you some of that data very briefly, but this is a tool that we're putting into very early trials to see whether we can impact neuroinflammation. There are still questions about the responsivity of this test and whether a successful therapy will move the biomarker, but it could be a very useful tool. I think digital phenotyping, for anybody who is at the American Academy of Neurology, would see that it's sort of in every aspect of what we're doing in neurology. If I have time, I'll show you some of the work that we're doing in ALS to try to develop more quantitative phase 3 measures about really, frankly, quality of life as well as function throughout life. It's not enough just to say biofluid biomarkers. There are numerous different fluid types that are relevant in any disease, but certainly in neurology. Cerebrospinal fluid is probably in neurology the biggest one that we think about as being sort of proximate to the site of action for diseases. And let me come back to that. We obviously get it through lumbar puncture. I'll put a pin in that and come back to it on the next slide. Blood, there are different components to blood, serum and plasma, which are different in that in one of them you've allowed it to clot. You can get rid of abundant proteins, but you might lose some of the non-abundant proteins. Might be easier to get exosomes from serum than plasma, but these are questions that you really have to ask yourself during this sort of biomarker process. Whole blood has uses for DNA and potentially epigenetics as well. We've looked at that in ALS a little bit and are developing some epigenetic platforms. And then peripheral blood mononuclear cells, which are, I think, maybe the most complicated biofluid-derived biomarker source to deal with. If you just want to look at cell types or counts, how many CD4 cells, that's not so hard, and you can shift blood in a multi-center study and probably get those counts very easily. But when we start talking about cell reactivity or RNA expression profiles, then the cells either need to be fresh or cryopreserved in a very specific way. That needs to be done on site and within a couple hours, and then you get sort of site-to-site variability. So it can be done, but it's expensive and really takes, I think, a lot of attention to detail to set that up right. Urine is a source of biomarkers. I think this has been a little surprising, but I'll tell you a story about one biomarker that's really developing nicely in ALS. And saliva potentially, I think that's maybe the lowest on the list for an obvious reasons. CSF is a rich source of biomarkers for neurology. Neurofilament that I'll talk about, it's more abundant in spinal fluid. Chitinases are only really tenable in CSF, as are many other markers that just aren't detectable in plasma, or their levels aren't relevant in plasma. SOD1, very common protein in the body, has a unique meaning in cerebrospinal fluid in certain cases. So there is SOD1 mutation that can cause ALS, or actually a number of mutations in SOD1 that can cause ALS. And in SOD1 ALS, SOD1 protein in the CSF may take on a particular meaning as a pharmacodynamic marker, not peripheral SOD1. Repeated sampling of the CSF is possible but challenging. We've run studies in ALS where we've gotten CSF as a part of an observational study every 4 months for years. People sign up for that. It takes a lot from the participant. It also is, I think, when you think about trial design, it takes a lot from the study team. You need a physician there doing a lumbar puncture, and that's costly and takes time, and you need to get study teams on board to do that. The implication for trials is really that you may not broadly be able to get CSF at multiple times from everybody in the study. So it might be a subset of patients that have frequent CSF sampling, or it may be that many patients have CSF sampled at a few time points. But I think it's probably folly to think everybody will have it, every 2 months, for example, throughout a trial. The last thing I want to say from a sort of very conceptual framework is that when we're thinking about disease monitoring biomarkers, it's important to think about how they fit into the pathophysiology, because that impacts their usefulness. And not only how useful are they, but in what scenario are they useful. So I think our mind tends to go to the canonical scenario where there's an upstream biological event which will eventually lead to the disease. There's a pathway of biology. Along that pathway is your biomarker, and then eventually that leads to the disease outcome. And if you measure a change in that biomarker, it really sort of inherently means that that it reflects a disease outcome down the line. That's how we all like to think about biomarkers that we're measuring. But especially early on, when we're just beginning to measure them, there are some other things that we have to think about that may be patterns. So it might be that there's an upstream biological event, and that is actually your biomarker. That's probably okay. In fact, it might be better in certain circumstances than having something that's along the pathway. But it does change how you think about that biomarker, and you need to sort of model that into your thinking about how you would use that biomarker in a clinical development program. Because if you're changing something that's further downstream, you might affect disease outcome without affecting your biomarker. It also might be that there's an upstream biological event that leads through a cascade to disease outcome, and that disease outcome then leads to a change in your biomarker. Again, that's not bad. It just means that you need to understand that you could change the biomarker without changing the disease outcome per se, and leads to a particular use of that kind of biomarker in your study. And then the last one that is even a little more complicated is that there could be an upstream biological event leading through some pathway to the disease outcome and through a separate pathway in parallel to a change in your biomarker. Again, that's okay under certain circumstances, but if you're looking to change your biomarker, you could change your biomarker and not the disease outcome if you're working on sort of the alternate biological pathway. And the thing that we really want to avoid is an upstream biological event, a cascade of events that lead to disease, and a change in biomarker that's completely unrelated. That would be an example of really a confounder, not a biomarker. But we have to be careful about that. So I want to put a little meat around this by using some very concrete examples from ALS. Neurofilaments I've referred to a couple of times. These are probably the most developed ALS biofluid biomarker. Again, not specific to ALS, and many diseases are looking at neurofilaments as biomarkers. So they're not diagnostic. It's really a disease monitoring biomarker that might have a role in clinical trials. These neurofilaments are highly expressed in neurons. They provide structural support to the cells. And there are three isoforms, light, medium, and heavy. And the light and heavy chain assays work best. They're most specific. The heavy chain actually undergoes a post-translational phosphorylation event. So when I talk about neurofilament heavy, I'm really talking about phosphorylated neurofilament heavy, if that makes any difference to anyone on the call. Just important to know that we're talking about that. Now, the level of neurofilament in the blood and spinal fluid mark neuronal injury, as I've said, and in ALS, they're elevated early and they're relatively stable over time. I'll show you some data to that effect, but I'll also show you the remaining questions. Because of these characteristics, they're very likely to be useful in early phase ALS trials to do what we said as a sort of a disease monitoring / pharmacodynamic biomarker, that is to de-risk programs. Here's some data just showing that both PNFH and NFL are elevated in ALS relative to disease mimics. Note that there is overlap. It's not a clean separation. That's true of most biomarkers in ALS. Important, and I'll come to what the implications for that are in just a minute. Neurofilament, this is light, it's true of heavy as well. It's increased in CSF and plasma. Do those elevations in CSF and plasma mean the same thing? Is it a passive diffusion? The levels are much higher in CSF than they are in plasma for both these molecules. The correlation is very high. I think the question we have to ask ourselves is whether these are outliers because of pre-analytic variability, analytic variability, or whether there's some different biology. I think we're honing in on the idea that really this correlation is strong, that we think they tell us the same thing, but we have to still be a little cautious about that when we start applying this to clinical trials. Neurofilament in CSF and plasma are higher and faster progressors. And again, this is neurofilament light data that I'm showing you, but the same is true for neurofilament heavy. And again, this is true, and the pattern is true in CSF and plasma, though the levels are much higher in CSF, and the separation may be a little better in CSF. So slow progressors are sort of spread out, mostly overlapping with controls, faster up here, and this other category is kind of medium progressors. And there's maybe predictably some overlap between these two. That means that there's some variability in this. It's not a perfect biomarker either. I think it raises the question, will we be able to detect change in slow progressors? And sort of the thing that throws that into even more question than we would say from the previous slide is that when we measure these longitudinally, the slow progressors in green have low levels and they remain low. They're not going up over time. So it's, I think it may be difficult for us to use this as a biomarker in slow progressors, and that might impact who we enroll into trials if we want to use neurofilament as a readout for an early phase trial. There's variability in the fast and the medium progressors, but they're all elevated. And if we could bring these down to low levels, I think we would have a convincing case that we should move a drug forward. The other thing is that I've said it, but here's some data to show that neurofilament appear to be stable over time in plasma, serum, and CSF. This is neurofilament light. The data are similar for heavy. But, not every study says that they're perfectly stable. So this is actually showing people who are both pre-diagnostic, who are gene carriers, as well as those people who have the disease, and it's neurofilament light in CSF, heavy in CSF, and serum, light and serum are the different colors. But you can see there actually is a trend downward in these markers over time, so maybe they're not perfectly stable. And if we look for just a very small change, we might mislead ourselves. So again, not a perfect biomarker. It means that we probably have to power to a bigger change than we might see, as a part of natural history of this. It would make sense if these are spilling into the spinal fluid in the blood from injured motor neurons, that at some point the steady state would decline as fewer motor neurons are turning over, because late in the disease there are fewer motor to lose. In animals, when we treat SOD1 ALS mice with the antisense oligonucleotide, we see that neurofilament is a good biologic readout. Now, because of the way that the mice work, we treat them while they're still developing, so neurofilaments in these mice actually go up over time rather than being stable. That's a difference in the model. We understand that. But the point is that we can blunt this increase in neurofilaments over time in the animals by treating with an antisense oligonucleotide. And I would say that at the American Academy of Neurology, Tim Miller, who's the lead investigator for the SOD1 antisense trial in humans, showed data that actually in humans, there is a decline in neurofilament in those who are treated with antisense compared to those who are treated with placebo. So we have some human data to say that this is bearing out in people as well. Neurofilament appears to increase in the year prior to phenoconversion. This dashed line is phenoconversion, and what we mean by that is this is where people who are gene carriers for ALS develop disease. And you can see that it goes up more dramatically after disease, but there is this slight increase in this year before. And we might be able to monitor gene carriers so that if their neurofilament begins to go up, we could enroll them in a pre-symptomatic trial, and that would be really a thrilling development. It'd be nice to detect it even further down the line, but 1 year ahead of time might be enough to stave off the disease. So to sort of summarize, much data suggests that neurofilament light and heavy reflect biology of motor neuron injury or loss, are relatively stable over time, may be responsive to treatment. For now, we might need to measure both as a part of trials, CSF in a select few, plasma and most, and sort of make sure that they pan out right. Neurofilament is not a surrogate endpoint at this point. So I think this is to de-risk programs early. This is to understand whether we're affecting biology. But the process for establishing for qualifying a surrogate endpoint is long. And really the current role is to help in phase 2 as we move to phase 3 trials. Urinary P75 extracellular domain is also exciting, but it's earlier in development. So P75 is on the cell surface of neurons. It's increased in neural development, and then it's downregulated later. And in adult life, it should be essentially undetectable, but it's re-expressed on injured motor neurons. And then it's cleaved off the cell membrane so that you can get this soluble P75 extracellular domain during apoptosis of those cells. P75 can be detected in urine. It is more difficult to detect it in CSF and plasma. It's there, but the assays don't work well. Urine has worked fairly well, at least in the hands of 1 or 2 labs that have done these experiments. So you can see in ALS there is overlap, but there's an overall increase relative to controls. And if we look at diagnostic categories, which essentially say how many regions of the body are affected, those with more broad symptoms of ALS have higher levels than those who have more restricted or those who are very early in the disease. And it's not as elevated in other neurologic diseases as it is in ALS. Now, following people over time in at least one experiment has shown that it increases over time. It's found in urine, and so when you do anything in urine, it's good to correct for creatinine. So here it's normalized to creatinine. And it goes up at a rate of about 0.18 nanograms per milligram creatinine per month. This needs to be replicated, and that's what I mean about it being earlier. But if we see something that's going up over time, it could be a very good biological readout to see whether we're saving motor neurons or slowing the rate of loss of motor neurons. The other thing is that it is probably also a prognostic biomarker, so lower P75 correlates with a longer survival, higher P75 in the urine, and this is extracellular domain, correlates with a shorter survival. And there may be a way to use that as well. However, if it's changing over time, this gets very complicated. So you'd have to know where people are in the disease and normalize to that. So that may not be its highest and best use. Now, it's only 1 or 2 labs that have been producing this evidence. There are more labs that are beginning to do it now. And we're also beginning to build it into trials in exploratory ways so we can learn more about it and get it to the point that it may be a useful biomarker more broadly. Data suggests so far that it does reflect biology of motor neurons and loss in ALS, that it increases over time, at least in the hands of these labs, can be measured in urine well, and it is at least theoretically possible that it could be treatment responsive. There is really no data to that end right now, but we need to start exploring that. Trials can include this very easily, and when you go back to sort of context of use, the downside to patients is almost none. We're collecting urine as a safety marker in most trials, and so it's easy to add it as sort of a learning stage biomarker or exploratory biomarker to trials now, and that gets us good data so we can understand it a little bit better. So that's sort of how I think biomarkers become exciting in a field, get incorporated into studies and into non-interventional studies and even interventional trials, and we understand what their role will be. Now, I mentioned earlier that pharmacodynamic biomarkers are becoming ubiquitous, nearly ubiquitous in ALS trials. And that, I think, is because of the negative phase 3 trials that we've had that, in retrospect, maybe if we had understood the biology better, we could have headed off of at least a few of those trials and put our efforts into something more promising. So pharmacodynamic biomarkers may help us sort of carry promising programs forward and stop the ones that aren't hitting their target. A few of the things that we're looking at, I've alluded to SOD1 and CSF. Regulatory T cell number and function in blood may be important, and I'll tell you a little bit about that. Markers of oxidative stress in CSF and plasma, and then I mentioned PET imaging of inflammation in the ALS brain as well. I think we're going to want to extend this kind of pharmacodynamic biomarkers. Some of them are disease biomarkers as well, some not, but we're going to want to include these in trials. So I showed you evidence about neurofilament in CSF. The other biomarker that's being used as a part of the SOD1 antisense oligonucleotide and gene therapy trials that are underway is SOD1 in the CSF. So this SOD1 appears to have a stable level over time in people with ALS. It's different from person to person, but within a person, there's a very stable level of SOD1. If we could use gene therapy or antisense therapy to knock down SOD1 and detect that in the CSF, that is really very close to a target engagement biomarker. And then neurofilament is sort of a more downstream pharmacodynamic biomarker that's also probably kind of a disease-related biomarker. It turns out that actually in that study that I talked about before, Tim Miller also showed that people treated with the antisense to SOD1 have had a decrease in SOD1 CSF protein relative to those treated with placebo. So this is working nicely. Those are early data in just a couple of patients presented just recently. Before we had that data, we had data from rat brain where the antisense oligonucleotide reduced SOD1 protein in rat brain and also reduced that protein in the CSF. So we had good reason to think that it would work in the trial if the drug is working. There are a number of trials that are targeting inflammation in ALS. One line of reasoning shows that people with ALS have lower peripheral blood regulatory T cells, and that those with more rapidly progressive ALS have even lower numbers of circulating regulatory T cells than those with slower forms of ALS that look more like controls, but not entirely like controls. There has actually been a phase 1 trial that we've been involved in during which we used plasmapheresis to extract T cells and then expand those T cells and give them back to people along with IL-2 to protect their phenotype. And it looked like, in a very preliminary way, like there may be some promise to that. We're now launching a phase 2 trial to build on that, and we can use this as really almost somewhere between a pharmacokinetic and a pharmacodynamic biomarker. What we're measuring in the trial is not so much directly related to disease, but more related to the actual therapy that we're giving to see whether we can boost and protect the level of regulatory T cells. And we'll have other pharmacodynamic biomarkers that we look at as well. We are also involved in a mesenchymal stromal cell secreting neurotrophic factor trial. So these are bone marrow-derived stem cells that are grown in culture and differentiated to secrete neurotrophic factors and then introduced back into the CSF. Brainstorm Cell Therapeutics is the sponsor of this. And it's in a phase 3 trial now. In the phase 2 trial, what we saw was that a number of, but not all, of the neurotrophic factors increased in those who were treated with the cells and not in placebo. And VEGF was one of the ones that was most affected, so was LIF. So this is a very nice pharmacodynamic marker to say we're at least doing what we think we want to do. We also looked at inflammatory markers in the CSF. MCP1 is probably the best developed, the most reliable in ALS. It's elevated in ALS, and we saw a decrease in that elevation in the stem cell treated patients, but not in the placebo treated patients. I've talked a little bit about PBR28 PET. So this is a tracer that looks for really activated microglia and perhaps some astrogliosis. In natural history studies, we can see that in healthy controls, we don't see any activation in the motor cortex, but in ALS, we do see an activation that is the sort of red-yellow color in the motor cortex of the microglia. We see that in PLS as well, which is a purely upper motor neuron form of ALS, very closely related, and we would anticipate that we would see it there. We're now beginning to bring this as a marker in a number of trials, early phase trials, to see whether we can impact inflammation, neuroinflammation in people with ALS. I think I don't want to focus on it too much, but I think we're going to need to sort of make some nod to computational medicine, artificial intelligence, and sort of models, and their role as ALS trial outcomes or tools in trials, almost like biomarkers or sort of computational biomarkers that can help with trial design. So in ALS, Origin Data Sciences and NCALS both have prediction models that allow us to really differentiate who will go on to be a slow progressor, a medium progressor, and a fast progressor. Now, they haven't been tested prospectively as much as we'd like them to be, and so we're beginning to incorporate them just in a learning way into trials. But if this pans out to be true, it has implications for how we enroll patients, stratify patients, and even sort of building it into our model could give us substantial statistical benefit over what we're doing now, and it could be a really important sort of prognostic biomarker. This is another computational model using a mixture of Gaussian processes to look at ALS trajectories and to say that we might actually be able to have not just 3 groups, but different patterns of disease that may give us even more benefit. And then we might even be able to identify subgroups that have much less variability. And these are actually clusters that are identified completely by a model, but when we go back, they map onto what we think are important variables for disease progression. And those are bulbar onset and the rate of progression pre-diagnostically. So how much change there is in a person before they get a diagnosis. So really interesting, I think, computational biomarker work that's going on. I want to talk for a moment about phase 3 trial efficacy outcome measures and what we're doing to try to improve the quantification of our phase 3 efficacy markers. And this really comes down to a central challenge for ALS efficacy trials. It's an uncommon disease, and for our best outcome measure, the ALS functional rating scale, in 2011, I think I sat down with David Schoenfeld, a very senior biostatistician, done a lot of work in ALS, and we really sort of worked out that it's very difficult to get smaller than 355 patients in a trial and be, very certain that you're looking that you'll be powered well for the kind of change that you might see in that trial, something like a 30% change in efficacy. This is sort of central to how we design trials. If we could get that number down to half or a third, it would be a huge benefit. We could run 3 times as many trials. The typical outcome measures that we've used in trials are tracheostomy-free survival. This is just survival, but some people choose to have a tracheostomy, and that's chosen as sort of an equivalent to survival, assuming that without a tracheostomy, they would be nearing the end of their life. Vital capacity, which is just the volume that people can breathe out in a single breath. And the problem there is that we're measuring really a group of motor neurons, specifically the ones that go to the diaphragm and the muscles of respiration. Handheld dynamometry is quantitative strength testing. It's a way of using a load cell to see how many pounds of pressure given muscles can generate. And then, as I've talked about, the ALS functional rating scale, revised ALS-FRSR. It measures bulbar function, fine motor function, gross motor function, respiratory function. There are 12 items and 48 points. On average, there's about a 1 point per month decline. there are challenges with each of these. Tracheostomy-free survival is definitive, but there's a low event rate over a relatively short trial, so we have to do long trials, a year to a year and a half. That's a long time in this disease. It's complicated by this tracheostomy issue, and a therapy might affect function or even quality of life without necessarily having a discernible effect on survival. Vital capacity is limited just to that population of motor neurons that affect breathing, and some people might not have any weakness there until later. Also, bulbar weakness, facial weakness affects the measurement of the outcome measure, so that's a problem with vital capacity. Handheld dynamometry tests all the muscles in the body, but it's difficult to summate. So some people might have leg weakness, some have arm weakness. How do you compare those? So it's sort of either too limited or too broad in scope in some ways. The other thing is that rater strength and patient effort can affect the results. So it's not perfectly objective either. And the variability we find is no better than our other outcome measures, and there are some ceiling and floor effects. ALSFRS-R actually performs best of all our outcome measures. It may give too broad a picture for some patients. It's subjective, so it bounces around depending on how patients are feeling, and we can see that. If you make them feel nauseous with a drug, even though it's not affecting ALSFRS-R, their function directly, it puts them in a bad mood, and they tend to rate it a little bit lower. It's also not clear what clinically meaningful change is, and it's hard to compare two people because they might have changes in different subscales that mean different things. And the other thing is it doesn't progress to zero, so analysis is complicated by death. We need to do sort of last value carried forward. So one of the things that we're doing to try to address this sort of phase 3 monitoring is disease monitoring biomarkers using digital phenotyping. And so this is sort of, in one form anyway, it's an app on a phone, but there are lots of forms of this. It's really the moment-by-moment quantification of individual level human phenotype in situ using data from personal digital devices. This is a definition from J.P. Onella, a partner at the School of Public Health on these projects. And we can divide this into two forms of data: passively collected data. This is sensor data, usage-based data. So you let people use their phone and you collect data from that and just see how they're living their life. They don't need to engage at all. There's more noise in that data, and we need to have sophisticated algorithms to overcome that. And then there's actively collected data. User engagement is required: surveys, tasks, voice recordings. And they have a different role, but are also very important. And we've done at least one study using this where we follow people for 24 weeks. We get in-person sort of data, our basic data, vital capacity, survival, ALSFRS-R, and a score I didn't talk about called the CBS. And then we have some active data, the ALSFRS-R at home, and a speech recording and a cough recording. And then we get passive data from the accelerometer and GPS. And I'll show you a little bit of this data coming out of this. One of the first questions we asked is really, does a self-entry ALSFRS-R, which is not how this questionnaire was meant to be used, does it correlate well with an in-clinic delivered ALSFRS-R? And you can see this is a pilot project. We don't see outliers. The correlation is very high. When you allow people to enter their own score, it's about 5% higher than clinic-based. So that seems to be true in this data set. We have some follow-on data sets where it also seems to be true. Very highly correlated, I think could be useful. I don't think I included this slide that shows its progression over time, but it progresses over time very similarly to our ALSFRS-R measured in clinic. So we might be able to get this data completely remotely, and that would reduce the burden of trials. Speech is actually a very good indicator of change over time in ALS. Looking just at pause time, which is only one parameter that we can get out of speech recordings, we can see that speech rate is actually increasing in all but one participant in this trial. Not all of them reported bulbar symptoms on our questionnaire, so we can actually detect these changes before patients can detect them themselves. And this may be a very sensitive outcome measure for trials, but we need to look at the noise. You can see there's some spread around those measures, and I think we need to understand how it will perform before we start making it a important endpoint in trials. So we're beginning to incorporate it now to see how it performs relative to our well-established outcome measures. And then I think the most experimental thing that we're getting out of this is really the GPS data. So in people in that pilot study who had a good baseline ambulation. That is, they were walking, and we determined that based on their score on the functional rating scale. There's a question about walking. We looked at their daily distance traveled using GPS, and that daily distance traveled actually declines over time. We looked at their time spent at home, and that actually increases over time. We're still, you can see that there's sort of, we're dealing with sort of the two ends of the graph here, and that has to do with sort of the processing pathway. So this is very early data. I would not take it as gospel, but what I would say is that it's an interesting way to look at the data. Interestingly, there's probably a floor effect here, and people who are not walking, we don't see a change in their daily distance traveled. It's less than what we see in the previous slide, but it's not changing over time. And their time spent at home really doesn't increase over time either. We're looking into what these variability that we see in some of these lines are too. Again, early, but this may give us a more quantifiable phase 3 outcome measure. Lots of work goes into a lot of these the things that I've shown. Many of these are multi-center efforts, and so I want to acknowledge that. And there's been support for some of these projects from many, many different organizations. So I'll end there. I guess I would wonder if there are any questions. I think I should end the show, and then maybe.
Are there any questions or comments from anyone? You can use the chat box if you don't want to speak up. Dr. Berry, do you see that comment? It says excellent talk, which I agree.
Oh, thank you very much. Yeah, I'm not sure where the comments are, so if you quite figure out what the comments.
Yeah, once you stop sharing, you can see the comments in the chat box. But.
Oh, I see. Okay, let's see. Stop sharing.
There is a question that says, is there any general advice for fields that haven't really started the biomarker endeavor yet?
You know, I guess so I've been working on this for a long time, and really we used to write grants that would say something like, you know, we're going to collect plasma and CSF, and we're going to look for biomarkers, and you know, then we'd have like a couple that we would incorporate, and then we'd say, and we're going to look for other ones. And I think that you know, it got us a lot of banked fluids, and that was helpful. I think you need, you know, if you're going to look for biofluids, you need biofluids. If you're going to look for you know, signal in data and other, you know, in electrophysiology or in imaging, you need those things. So one thing I would say is establish some parameters for sharing and a collaboration, because I think that will get you a long way. I think it's harder now to write biobanking grants to almost any foundation. Maybe disease-specific foundations are still open to that, because I think they see how it can be important for that specific disease. I think now if you can guide, even though the outcome, you know, what you're really trying to do is collect and bank some fluids, if you have a lead biomarker and you can put that into the context of those documents, the qualification document and the BEST document from NIH and FDA, I think that goes over really much better with reviewers to have some specificity around what kind of biomarker you need, why do you need that, how will that move the field forward, and why is this a good putative biomarker.
In your experience, what grant funding mechanisms best fund biomarker studies?
So for a while, NIH was actually doing a good job of funding these. I think that they've backed off on at least banking and sort of clinical data collection without a more directed effort. I think if you have a biomarker that is your candidate biomarker and you lay out a plan for how you'll develop it, I actually still think NIH is a very reasonable place to go. But before you get there, you might need to go through other sources. The two places I would say are really good are disease foundations. So if you have a disease that has a good foundation, we have ALS Association, as well as Muscular Dystrophy Association, and a few smaller organizations, even ALS1, Project ALS, sort of more regional organizations, that you can sometimes cobble together funding for a bigger project to get enough data to go to NIH. And the last thing I would say is that industry is actually not a terrible place to go. There are a lot of potential industry partners that need some kind of biomarker to de-risk programs if they're going to get into your disease area. And they might be willing to at least partially fund it. If you could bring some philanthropy, some foundation money, some NIH money, that might actually lead to a great collaboration.
Are there any other questions or comments? We have a couple more minutes. Okay, so it doesn't look like it. So I appreciate, Dr. Berry, you taking the time to present to us today. Oh, wait, there's one more here. NIH is interested in validation of biomarkers, not discovery.
Yeah, I mean, I think, it's a good point. I think that's why I sort of say sometimes you need to get to a point where you have enough preliminary evidence to go to the NIH. It's like anything else with the NIH. And so you might need to kind of, use some of these other avenues to get to a point where you have a biomarker that is, maybe it's not just validating it, but you need to at least have shown that it's a good candidate biomarker, I think, by the time you get to the NIH. I agree with you, and I sort of wish it weren't that way, but I think that's largely true.
Okay, well, thank you again for presenting today. I just want to remind everyone that's still on the call if they could just evaluate the webinar. The link is in the chat box here. And that our next webinar is June 25th at noon Eastern. So thank you, everyone, for joining. I appreciate it. We'll see you next time.
Thanks so much, Joy. Take care, everyone.
All right, take care. Bye-bye.